notes that we've done already in class. It's called Things You Already Know, Lab Safety and the Scientific Method. The first thing you'll see on your lab paper is some lab safety symbols. This one is called biohazard. A biohazard means organisms or other biological materials that might be harmful to humans. If you ever go to the doctor and have your blood taken, they'll put the syringe in a container that has this symbol on it. It's called biohazard. That's short for biological hazard. Flammable material is this symbol here, with the flame and the red diamond. Some materials catch fire easier than others. Usually, if you see this symbol, it's on something very flammable, like a tank of gasoline or something. Risk of electric shock. Electricity flows through conductors and is stopped by insulators. Your body is a conductor, so your body will conduct electricity if you give it a pathway. So if you see this symbol, don't touch whatever it is. The skull and crossbones means toxic or poisonous. Poisonous materials should not be ingested or eaten. This symbol means chemical hazard. It indicates chemicals that can react with or destroy tissue and other material. Basically, if a substance can give you a chemical burn, it might have this symbol on it. And animal safety. It's used when the safety of animals and students must be ensured. Radioactivity. Some materials are unstable and emit radioactive particles, and they would have this symbol on them. So some kind of nuclear device or nuclear waste might have this symbol on it. And extreme temperature. If you see this oven mitt, it could be an extremely hot or extremely cold substance, either of which could burn you. So for each of those symbols, you should have what it means, just the name of it. You don't need the little description, but just with the name of it. Now some general safety rules for the lab. Read instructions before attempting to do anything. When instructed to do so, wear gloves or goggles. Notify your teacher if any spills or accidents occur. No food or drink in the lab, including no chewing gum. Keep your work area uncluttered or clean, organized. Know where to locate the fire extinguisher. In my room, it's right by the door. Never put anything into your mouth during a lab experiment. Clean up after yourself. No running, horseplay, or practical jokes in the laboratory. Broken glassware should not be disposed of in a classroom trash can. Notify a teacher for special instructions. Follow the instructions of your teacher when disposing of all chemicals. Wash your hands after handling hazardous chemicals. After that, it's the scientific method. First, you state a problem or ask a question. Next, you conduct observation and research. Then you formulate a hypothesis. Then you conduct your experiment by collecting data, analyze that data and summarize your results, draw a conclusion, and finally communicate your results. So for the most part, those things need to be in that exact order. Observation and research, that could begin before you state your problem and ask a question, but for the most part, you can't conduct your experiment without a hypothesis. You can't analyze your data if there is no data, and you can't draw a conclusion unless you have results, etc. Science vocabulary. Variables are things in an experiment that change. The independent variable is the factor that is intentionally varied by the experimenter, also called the manipulated variable. The dependent variable, also called the responding variable, is a factor that may change in response to the independent variable. So if you have an experiment where you're feeding plants things besides water to see how they grow, you'd have a plant that you were giving water to, a plant that you were giving some other liquid to, perhaps coffee or cola or something like that. Um, the independent variable would be the liquid that you're changing. and then the dependent variable would be how tall the plants grow. Usually on a graph, or almost always, your independent variable goes on the x-axis and your dependent variable goes on the y-axis. Your control group. In a scientific experiment, the control is the group that serves as the standard of comparison. The control group may be a no-treatment group or an experimenter selected group. So if you've got your plant experiment and you've got plants that you're giving water, plants that you're giving Coke, plants that you're giving lemon juice, plants that you're giving carbonated water, the control group would be the group that gets regular water, just kind of the no treatment group. You're not really altering them from their natural conditions. Trials refer to replicated groups that are exposed to the same conditions in an experiment. Basically, you're running the experiment multiple times, either concurrently or one after the other. In an experiment, the more repeated trials you can include, the more reliable or valid your results become. Mass, volume, and density. Mass is how much matter an object contains. Volume is how much space it takes up. And density equals mass divided by volume, or D equals M over V. And volume by displacement. To find the volume of a solid, especially an irregularly shaped solid such as this diver, you would start with a graduated cylinder that had a known quantity of water in it, and this graduated cylinder on the left has 3.4 milliliters of liquid inside, or water inside, and then you add the thing that you want to measure the volume of, and you see the water goes up. So it went up to 5. So if you subtract the final volume minus the initial volume, you're going to get the volume of the thing that you added. That's called water displacement. In this example, the diver's volume is 1.6 milliliters. That's 5 milliliters minus 3.4 milliliters. And so you can watch the video again if you need to, pause it if you need to, and you get the notes from me in class. All right, thanks.